Hello, I am Preethi BC, first year MSc Biochemistry, and my topic is DNA sequencing by Sanger method. DNA sequencing by Sanger method is also called as chain termination method. It is a method for determining the nucleotide sequence of DNA. DNA sequencing by Sanger method. The standard DNA sequencing technique is the Sanger method, named for its developer Frederick Sanger, who shared in 1980 Nobel Prize in Chemistry. This method begins with the use of special enzyme to synthesize fragments of DNA that terminates when a selected base appears in a stretch of DNA being sequenced. These fragments are then sorted according to the size by placing them in a slab for polymeric gel and applying an electric field, a technique called electrophoresis. Because of DNA negative charge, the fragments move across the gel towards the positive electrode. The shorter the fragments, the faster it moves. Typically, each of the terminating bases within the collection of fragments is tagged with the radioactive probes for identification. Then example for DNA sequencing. Okay, consider the following DNA sequence. Draw the sequencing gel pattern from a result of sequencing the following template. The DNA with the DD and TP as the capper. So solution. The DNA template runs in 5' to 3' direction, and the synthesized DNA runs from 3' to 5' direction. The DNA sequencing, one more example is gel pattern type, where the W indicates the well position and the band that denotes the DNA bands on the sequencing gel. And this is the sequencing gel. This picture is a radiograph. The dark color of the lines is proportional to the radioactivity form. Phosphorus 32 labeled adenosine is transcribed DNA sample. And the reading of sequencing gel, it begins at the right, which are the smallest DNA fragments. The sequencing that you read will be in 5' to 3' direction. This sequence will be exactly the same as the RNA that would be generated to encode a protein. The difference is that the T base in DNA will be replaced by U residue. As an example in the problem given, the smallest DNA fragments of the sequencing gel is in the C lane, so the first base is a C. The next largest band is in the G lane, so the DNA fragments of the length 2 ends in G. Therefore, the sequence of the first two bases is CG. The sequence of the first 30 or so bases of the DNA are as follows. Then this is the codon table. The specific codon will code for specific amino acid, and the first amino acid will code for methionine, that is AUG. Translating the DNA sequence, the order of amino acid is in any protein is specified by the order of nucleotide bases in the DNA. Each amino acid is coded by a particular sequence of 3 bases. To convert a DNA sequence, first find the starting codon. The starting codon is always codon for the amino acid methionine. This codon is AUG in RNA. And in this particular example, the next codon is AAG. The first base is A, so select the third major row of the table. And the second base is A, so select the third column of the table. And the last base of codon is G, select the last line in the black color of 4. Next, translating the DNA sequence. This entry AAG in the table is lysine. Therefore, the second amino acid is lysine. The first few residues and their DNA sequence are as follows. And methionine that AUG that code for methionine, AAG that code for lysine, and so on. This procedure is exactly what cells do when they synthesize protein based on mRNA sequence. The process of translation of cell occurs in a large complex called ribosomes. And this is the automated procedure for DNA sequencing. Here, a computer readout the gel generates a false color image where each color corresponds to a base. Then the intent sites are translated into peaks that represent the sequence. This is high throughput sequencing capillary electrophoresis. The Human Genome Project has spurred an effort of develop faster, higher throughput and less expensive technologies of DNA sequencing. Capillary electrophoresis separation has many advantages over slab gel separation. Capillary electrophoresis separation are faster and are capable for producing the greater resolution. Then capillary electrophoresis instrument can use tens and 11 hundreds of capillaries simultaneously. And the figures show a simple capillary electrophoresis setup where the fluorescently labeled DNA is detected as it exits the capillary. Thank you.